So in a normal year, we perform a similar experiment to part B in that we're looking at diffusion and osmosis in a system. In part B, we looked at artificial cells. Now we're going to look at living tissue. So the model organism for our osmosis study is a potato. So you have variable, different types of potatoes. Anybody know what kind this is? Nope. A russet, yeah. So it's there's sweet potatoes, there's red skin potatoes, yeah. So it's like a normal Idaho russet. And when you think about it, this potato compared to any other potato in the bag, is it going to be identical? Why not? Even if it's the same kind of potato, why not? Shape and size, and whatever condition that potato was grown in may have been variable. Like, it's not like all the potatoes that are in one bag came from like the same plot of land. It's just this massive collection of potatoes. So I'm going to ask that you go through and you look at the lab. It's the same idea as the artificial cell lab, except you know, in the artificial cell lab, you have different concentrations of sucrose placed in the bag. And then in the first case in water, and in the second case in 0.4 molar sucrose. In this case, you're punching out samples of the potato and you're putting them into a mystery sucrose solution. So that is, they're labeled A through F. They're different colors to kind of throw you off a little bit. But each one of those can be one of the following concentrations. It could be 0, 0.2, 0.4, 0.6, 0.8, and 1. Now, you don't know what each of those solutions is, but you should be able to experimentally arrive at that based on the rates of, primarily the rates of osmosis into and out of the cell, the cells, the collection of cells in the tissue, right? So, and then we'd break you up into different groups, and some of you would do one kind of tuber, one of you would do another kind, and then we'd compare results and talk about why we got those results. So obviously we're not going to do that today. I'll show you how the experiment is usually performed, but here's a quick walkthrough. You take core samples out of the potato. Ideally, you want to use the same potato. So you would take this thing, literally put it down onto the table, push this thing through. It's kind of cathartic, right? Kind of fun. And once you do that, if you push this other smaller tube through, you get a core sample of potato. So it's potato tissue that we're doing our experiments on. Now, I would then take this and put it into a weigh boat. Think about why that is. Why do I want to put my sample into a weigh boat, not on a paper towel? I also want to take off these edges, right? The skin of the potato. So I would take a scalpel or a razor blade and cut that off. And this would be basically what I'm experimenting with, a tube of potato cells. So the way that we would do this here is we put that onto a scale, measure the mass of your samples, pre-exposure to the unknown mystery solution, put them into the samples overnight, and you come back in the morning, blot off the excess fluid, and do a second weighing that gives you the final mass of your sample, okay? So we're not going to do that though. It's actually kind of fun and cathartic to do it. I'm going to just give you the data. Okay, so now it's the next day. You've gone, you've measured before, and you've measured after. I'll still ask you to calculate percent mass change using the same formula as before. And then I'm going to ask you to do some graphing. So here's the information that you can use. You go to this link right here. And these are all the questions I want you to work on today. Okay, so go through and consider this. as with all the labs in here, I want you to draw out a graph showing your data. So what does that data look like? I give you some pointers on how to consider graphing that, but essentially you're going to try to guess which of your data points is related to like being 0, 0.2, 0.4, 0.6, or 0.8 or 0.1. You should be able to infer that based on your knowledge of osmosis. Okay, so take a look at the instructions. We're going to work on this virtually for the rest of the class. In addition to drawing a graph and answering some of these questions, I'd like you to write a 2-paragraph lab report. So again, it's not like you have to write a whole lab report. I just want you to discuss the graph that you created. Why did you guess, not guess, predict that, I don't know, beaker B or solution B contained, 0.6 molar sucrose solution? Why did you say that? Why did you say this one was 0? Why did you say this one was 0.2? Are there any questions on that? Any questions at home?
So for the like lab report, besides like the what you just said about like how, what, and like why you decided, like, do we need to like kind of like follow kind of the directions that like book that like we had to read over the summer, like what they said was in a lab report, or it's just kind of like paragraphs explaining what we did or anything?
So to be kind and fair in AP Bio, I will never ask you to write a full-on lab report. So yeah, you're absolutely right. In a research paper or a high school lab report, you'd write an abstract, an introduction, materials and methods, you'd show your data, you'd discuss your data, and then you'd conclude something. To help save you time and energy, since I just basically gave you all that stuff, the background and the materials and methods, I'll just ask that you draw the graph and then use your knowledge and reasoning to explain to me why you guessed, which sample was 0.2, 0.4, 0.6, and etc.
Okay.
There are some clues in the paper too. So when you get to these documents, just make sure you read through everything, because I'm starting you off in certain directions, and I'm asking specific guiding questions to help you write it. Okay? And for you guys at home, I'm going to let you work on this asynchronously. We have our next class is planned to be a review catch-up day. So these will not be due, the graphs and the 2 paragraphs, until the class after next. So the class where we start our discussion on metabolism and talk about energy, I'd like for you to have those paragraphs to me by then, and the graph. So we have some time to do it. That's not to say, go waste this class. You should get a good chunk of this done during this class, but it's not actually due until after next class. Is that clear for everyone?